Before you learn the structure of antibody, you must know how antibody was discovered and why antibody is called gamma globulin. Actually, antibody is called immunoglobulin. So why it is called gamma globulin? In this slide, I will explain an experiment performed by Kabat. He performed a simple experiment. He injected or immunized a rabbit with ovoalbumin protein. After, he took serum from that immunized rabbit and he make the serum into two aliquot. Aliquot means just to separated the serum into two portions. From the first portion, he performed gel electrophoresis and he found 4 peak of protein. He knew that the highest peak is albumin because in our serum, albumin present very high concentration. Then he found another peak. I am talking about the blue line. This blue line is the first gel electrophoresis experimental data. So he found albumin peak. Then he found 3 other peak. He called it alpha, as it is the letter of Latin, alpha, beta, and next he called gamma. Then he performed an interesting experiment. He knew that antibody is responsible for precipitation. It is called immunoprecipitation. Antibody can bind with antigen and can precipitate. So he wants to know which portion of this protein is actually antibody. So he performed a simple experiment. He added ovoalbumin in the second aliquot of the serum and he found that there is no fall of albumin. So albumin is actually not the immunoglobulin, but there is slight fall of alpha and slight fall of beta. So this black line is showing the second aliquot experiment where ovoalbumin was added. He found a slight fall of alpha protein and beta protein, but a huge fall of gamma protein. You can see the peak before adding the ovoalbumin was there, and now after addition of ovoalbumin it falls into this protein that could be antibody. So he called it gamma globulin. So that's why antibody is called gamma globulin. So in the serum protein he found that the gamma globulin is responsible for the binding of antigen, and by binding the antigen like ovoalbumin, it precipitates out. So the protein now falls. The level of the protein falls. In this slide it is showing the antigen antibody complex. The first is zone of antigen excess. Antigen concentration is high, so there forms small complex. And in the right side, zone of antibody axis, the concentration of antibody is high than the antigen. There also small complex form. The antibody can bind with its 2 arms. It form large complex, and this situation is called zone of equivalence. In this situation, a antibody antigen can be precipitated out. So now you can see the structure of antibody. Antibody is mainly composed of 2 chains called light chain and heavy chain. In antibody, there are 2 light chain and 2 heavy chain. The light chain composed of 2 regions called variable light and constant light. And also the heavy chain contains 2 portions. One is variable heavy and constant heavy. But there is a difference. The light chain contains 1 variable region and 1 constant region, but the heavy chain contains 1 variable region but 3, or in IgM antibody the constant region is 4. This variable heavy and variable light chain make a region called that is responsible for antigen binding, and the constant region is responsible for the effector function of antibody. There are some exceptions like human or other vertebrates contain heavy chain or light chain, but camelids and the shark do not have light chain. An experiment by which scientists have known the structure of antibody. In the first situation, scientists have digested the antibody with an enzyme called papain that is found in papaya. This papain enzyme can cut and form 2 separate fragments and a separate constant fragment. This fragment is called FAB because it means fragment of antigen binding. But when antibody is digested with papain, FAB, single FAB cannot bind with antigen, and this region is called FC region because fragment of crystallizable, because it spontaneously crystallize. And if the antibody is digested with pepsin enzyme, the enzyme cuts the antibody here so that 2 FAB can join and the rest of the Fc portion is digested. So one segment was found called FAB bar 2. In this case, I want to mention this FAB, single FAB cannot bind with antigen, but this FAB bar 2 can bind with antigen. In this slide, I want to mention that not only the antibody falls in the immunoglobulin family, but there is a super family. B-cell receptor also falls in the immunoglobulin family. Class 1 MHC molecule and class 2 MHC molecule falls in the superfamily of immunoglobulin, CD4, CD28 and some adhesion molecules like ICAM, VCAM. Next is the isotype, or it also called class of antibody. All the class or isotype of antibody actually differ only in the constant region. So they perform different function. As I have already mentioned in my previous slide that the constant region is responsible for the effector function of antibody. So all the antibody perform distinct function, and all the constant region of these 5 antibody are represented by different segments of gene in case of mice or human and any other animals. In this slide, I want to mention some important functions that are performed by antibody. Like IgA antibody develop immunity in the mucosal area. IgD acts as a B-cell receptor. IgM also acts as a B-cell receptor and complement activation. IgE develop immunity against helminth and also have a negative point. It can produce or generate immediate hypersensitivity like allergy, hay fever, hives, asthma and eczema. IgE antibody have many different functions like opsonization, complement activation, ADCC, neonatal immunity. In my different classes, I have explained what is opsonization, what is complement activation. ADCC means antibody dependent cell mediated cytotoxicity. The antibody composed of a region called hinge that gives it flexibility. So you can see widely spaced cell surface determinants it can recognize, and also closely spaced surface determinants can be recognized by same antigen. So there is a flexibility by the hinge region. There are 2 types of antibody. One is membrane bound condition. The difference you can see just in the transmembrane domain of these 2 antibody. In the secreted form of antibody, there is no transmembrane domain or transmembrane region.